and welcome to the Roche Tissue Diagnostics Discovery webinar. My name is Mark Helliwell and I'm the International Product Manager for the Discovery product portfolio at Roche Tissue Diagnostics. I'll be your moderator for this webinar, which is titled Combining the Discovery Proximity Detection with Chromogenic Multiplex, Discovering the PD1-PDL1 Interactions. Discovery proximity detection is a powerful new tool that extends the capabilities of traditional IHC assays, enabling in situ detection of protein interactions and post-translational modifications with high sensitivity and specificity. By combining this technology with IHC multiplexing, we enable researchers to gather valuable information with respect to signaling pathways and predictive responses to therapies, all within the context of tissue. The recent advances in bright field multiplexing on Discovery Ultra staining system has allowed researchers to easily access these complex testing methodologies. So before we get started, please note some technical aspects of today's webinar. This will be available on demand within the Labroots platform and also via YouTube. And we encourage you to ask questions. To do this, please type the questions into the questions tab and we'll make sure that our speaker will address them after the talk. And please feel free to leave feedback and your ratings. Your input is highly valued and will help us bring the best educational programs to you. So now I'm pleased to introduce today's speaker. Dr. Thierry P.P is a postdoctoral researcher at the Erasmus University Medical Center, Rotterdam in the Netherlands. In 2014, he graduated from the University of Applied Science in Leiden and specialized as a pathology technician. In 2018, he finished his PhD at the Erasmus University Medical Center on the subject of monocytes and macrophages in solid organ transplantation. After finishing his PhD, he enjoyed the pathology and research trial service, where he optimizes and introduces new technologies within the field of pathology and supports scientists on specific scientific questions, especially in the field of chromogenic and immunofluorescent multiplexing. Thank you for being here, Thierry. I'll now ask Dr. P.P to provide some insight into his research with proximity detection and IHC multiplexing on Discovery Ultra.
Thank you, Mark, for this very kind introduction. And I will start my presentation on combining the discovery proximity detection with chromogenic multiplexing, and then be focused on the discovering of the PD1-PDL1 interaction. So here's a disclosure slide where the proximity detection kit was supported by Roche Tissue Diagnostics, and we used the demo version 4 of the discovery proximity detection, which was provided by Roche Tissue Diagnostics. This is for research only and not for diagnostic purposes. And this presentation is sponsored by Roche. So I want to start with some background and utility of the PLA. And the discovery proximity detection kit is for use on the Discovery Ultra platform, which you can use completely automatic. And what Mark already just now said, it's a kit which you can study the interaction of two unique proteins of interest in situ. But you can also use this to exclude cross-reactive binding and detection of irrelevant proteins, also called off-targets. And the importance of this PLA technique can be said is estimated over 80% of proteins are functioning in complexes to elicit biological responses. So the interaction of proteins are very important, and what is better to visualize them. What makes it even better is if you add chromogenic multiplexing. Chromogenic multiplexing is a technique where you can use multiple biomarkers with different colors together to study the marker colocalization and its microenvironment. In this way, you can study different cell populations like you do with flow cytometry, but then in your tissue. When you add this to the proximity detection, you can already think that you can find the cells who are interacting with the different receptor and ligands. So the proximity ligation assay was firstly developed and described by Fredrickson et al. In 2002, where they used an analogous technique where they did a ligation of oligonucleotides, which eventually gave rise to an amplifiable DNA sequence. 10 years later, approximately 10 years later, the PLA became a generally recognized tool for advanced and precise protein detection, also using immunohistochemistry. The PLA enables exceptional specificity and sensitivity of protein detection and quantification of immunocytochemistry, but also immunohistochemistry applications. And this presentation, of course, involves the Discovery Ultra staining system. So what is known about the PLA? What has it done before? What is the clinical value of this technique? So PLA has been studied in a number of studies already, where they both studied EGFR dimerization and receptor activation, which has been proposed to play a crucial role in tumor progression and development of drug resistance. And in this kind of study, they looked with PLA to receptor dimerization events with mutation-specific PLA, and it appeared to be more suitable for the selection of patients who have EGFR-targeted treatment. They also studied this in breast cancer, where elevated levels of HER2 or HER2-HER3 complexes were detected with the PLA, and they showed significant association with decreased recurrence-free survival and a reduction in overall survival of breast cancer. So being able to study primary tissues or tissue samples and cell lines with PLA can give valuable information of signaling status and prediction of the response to a certain therapy. There's another study which was in 2015 where they studied glioma samples, and in this study they approached the question of whether IDH1 epitope is presented on HLA-DR in human glioma tissue. And to this end, they used proximity ligation assay, which until now was used as a tool to visualize endogenous protein-protein interactions or fusion proteins in situ, and adopted the technique to analyze binding of IDH1 derived peptides to MHH class II molecules in vitro and in situ, just to show you that PLA can be used also in different ways. So in this figure, they show the specific co-localization of IDH1 peptide and MHH class II in primary glioma cell lines. And later, they also validated this in glioma tissue of patients, where they used the PLA technique and found specific co-localization of IDH1 and MHH2 in glioma tissue. And in this way, they show the importance of this technique. But to come back to the specific topic of this talk, we're going to discuss in more detail a recent paper our group published in Cancer Cell, where we investigated the PD-1, PD-L1 checkpoint or pathway using the PLA. And I will not discuss this paper completely in detail because that will take too long. I will focus on the tissue and the proximity ligation assay technique. So for the background information, there were several recent insights in exploring the biology of the PD-1 receptor and its corresponding ligand PD-L1. It offers clues into what drives the ICB, which means immune checkpoint blocking drugs efficacy. Besides tumor cells, myeloid cells expressing PD-L1 have been revealed to be essential for this ICB efficacy as an anti-PD-L1 antibodies remain effective in transplanted tumor cells lacking PD-L1 in a variety of models and studies. These discoveries offer explanation for the rather unexpected finding that PD-1 primarily acts by inhibiting signaling downstream of the CD28 co-stimulatory receptor following B7 ligations, which supposedly was by myeloid cells. Where this interaction and therapeutic disruption in case of PD-1, PD-L1 takes place is still unknown. So the aim of this study was to find PD-1, PD-L1 proximity detection is to investigate the role of tumor-draining lymph nodes in efficacy of anti-PD-L1 therapy in multiple preclinical tumor mouse model and in human tissue. And for this talk, we focus on the human tissue. So just to have the basics of this PD-1, PD-L1 interaction, what happens in the tumor is there's a tumor cell which has a specific antigen and it releases pieces of the tumor which has an epitope, and this epitope is then recognized by the antigen presenting cell, which presents this antigen using the PD-L1 to the PD-1 on the T cell, so that the T cell can release cytotoxins inducing apoptosis in the targeted tumor cells. So now I will show you what the discovery proximity detection reaction looks like. When you have your tissue slide and you want to investigate the two proteins, the PD-1 and the PD-L1, and whether they are interacting, you use the primary antibody, which has to be a mouse and a rabbit monoclonal. So in this case, it's a mouse anti-human PD-1 and a rabbit anti-human PD-L1, clone SP263. You detect this with a secondary antibody in the case of PD-1 with an anti-mouse AP labeled secondary, and in the case of the rabbit anti-PD-L1, it's an anti-rabbit caged NP. So this NP molecule is caged. When the targets are closer than 40 nanometers from each other, so which means they are interacting, the cage will be uncaged and can then be detected with an anti-NP HRP labeled antibody, which then can react to any color you like to choose. As you know, in the discovery platform, you can use different colors, which are HRP based. In our case, we use DAB, but you can also use a red, green, silver, blue, teal, or purple. So how to start with this protocol? Most important is to figure out your antigen retrieval step. So for the discovery proximity detection, you use the universal procedure in which the antigen retrieval step is depending on your antigens of interest. In case of our study, it's PD-1 and PD-L1, which uses a CC1 for 64 minutes at 97 degrees Celsius, or in their case 95 degrees Celsius. Here you can see how that looks like in the procedure. So you click until you have the CC1 64 minutes selected. Of course, you should not forget to do deparaffинation before the antigen retrieval in case you're using FFPE materials, and when you use frozen sections or cytology material, you don't have to do that. You can load it as a wet slide. So after you use the discovery inhibitor CM for 12 minutes, which is a peroxidase inhibitor, you can select the antibody and you use the primary antibody co-incubation function. In case of PD-1 and PD-L1, we incubate both of them at 60 minutes at 37 degrees. That looks like this in the procedure. You click on antibody, you click on primary antibody co-incubation, select the temperature of 37 degrees Celsius, and then you click on which antibody you use, and in our case it's PD-L1 SP263 and the PD-1. It will then add 1 drop of both antibodies on your slide and it incubates for 1 hour. Then we'll start with the detection of this after this primary antibody incubation, and then you select on the enzyme conjugate and you use the anti-NP-HRP for 12 minutes. Then you can amplify your signal using the discovery amplification step. Oh, sorry. And you need to choose the discovery anti-HQ or HRP for 12 minutes. So after that you can detect with the color you like. So after the proximity event, you can immediately go to the detection phase. Any color which involves HRP can be picked. In our case, we selected DAB. But of course, you can also choose what I also already said, red, green, silver, blue, teal, or purple. But please keep in mind, with the other colors, you need to give an amount of time and follow the instructions of Roche to see which time is good for your step. Then we do a counter stain with hematoxylin 2 for 4 minutes and a bluing step of 4 minutes. And I choose 4 minutes because then the background and the staining of your slide is a little bit less, so the signal of your PLA will be more clear. So in our study, this is what we found. Here you can see the signal at 40 times magnification in a lymph node of the PLA reaction. It looks a little bit like a stripe, which is between the cells, some dots. And this is how it looks like when you visualize this by PD-1, PD-L1 interaction. And when you quantify this, you can find, we counted these lymph nodes by high power fields. And what we found is that the average number of contacts in patients with an early recurrence of disease following surgery and no recurrence after 96 months was a significant difference. So this made us very interested in what kind of cells are doing this interaction, and we had to make a choice of which biomarkers we should investigate and which one we should add after the PLA. So how to gain insight into which cells were involved in the PD-1, PD-L1 interaction. So we investigated 3 additional markers using the chromogenic multiplex method. And we choose to investigate CD11C. We choose this one because this one is expressed on dendritic cells. Of course, it's also expressed on macrophages. And we also choose the marker for macrophages called CD68. And we wanted to know if the cytotoxic T cells are doing the interaction with one of those APCs. So adding markers for chromogenic immunohistochemistry. Please, you have to keep in mind that the discovery proximity detection can only be placed as a first sequence. So if you have a marker which has no pretreatment or a marker which is very sensitive to pretreatment, you need to maybe choose another antibody for this case because the first step always involves the PLA reaction. You cannot put it as last sequence. In our case, the discovery proximity already had a 64-minute CC1 pretreatment. We have to keep in mind. And we use DAB as a first signal for the proximity, and that is because we choose according to the color sequence. So this color sequence you can see as follows. You have silver, which you can use as a first detection or as last. DAB should be one of the markers you start with. Otherwise, you can get umbrella effect on your tissue. Purple can be next, red, teal, and yellow. If you follow this sequence, you can build up your chromogenic multiplex. And the markers we would like to add were CD11C, which is, in our case, a mouse antibody species, which is localized in the cytoplasm, and it can localize with CD68. And the single DAB staining had a CC1 of 64 minutes, and we want to use yellow as a chromogen. So we are going to put this marker as last sequence. CD68 was also a mouse species, which comes to expression in the cytoplasm membrane, and can also localize with CD11C, of course, and it had a CC1 pretreatment of 32 minutes in a single DAB procedure. So we're going to put this one as first after the PLA reaction with purple. And following the CD68, we want to investigate CD8, which is a rabbit monoclonal membrane marker, didn't co-localize with these two markers, and also has a CC1 of 32 minutes normally, but we want to use teal and put it in the middle. So you go back to your PLA procedure, which is optimized with your DAB detection, and you click on the dual sequence. When you use dual sequence, you need to do an antibody denaturation, and I would say always use this step with CC2 for 100 degree at 8 minutes. Eventually it will go for 24 minutes, and you have to keep this in mind because it counts as a sort of antigen retrieval step. That's why we make this tree of pre-treatment from low to high. You select antibody. In our case, we start with CD68 for 32 minutes, and we come back with a multimer HRP, which is in our case the OmniMop anti-mouse HRP for 16 minutes. And what you get then is, this is a picture of the complete PLA already, but what this color, what I want to show is that in the middle of this cell, there is a purple reddish signal, which is CD68. And here in the red, it's also CD68. It's red because here it's already overlapping with the yellow. That's why it became red. Then you start your third sequence. Your third sequence, you do an antibody denaturation again for 8 minutes. You click your third primary antibody incubation with CD68 for 32 minutes, and you come back with an OmniMop anti-rabbit HRP for 16 minutes. What you get then, it's a little bit difficult to see on this picture, but it's the teal signal of CD8 positive cells. Then you come with your fourth sequence, and you start again with the antibody denaturation step. And here, because our antibody in this case need to be pipetted on the slide, we use a manual application, our CD11C, and you come back with a mouse NP for 32 minutes. And you use the multimer anti-NPAP because we use Discovery Yellow, which works according to the alkalic phosphatase method for 44 minutes. And eventually you'll get cells who have the yellow cytoplasm stained, and when they co-localize with CD68 they become red, and you can see the brown signals of the PLA in between. So in the study it looked a little bit like this. We have these lymph nodes of these patients who had a melanoma, and these are the tumor draining lymph nodes, where you can see again in yellow the CD11C, in teal CD8 positive cells, and in reddish purple CD68, and brown the PD1 interaction. And as you can see in the germinal centers of these lymph nodes, there were a lot of interactions happening, but also outside of the lymph node follicles, there were also a lot of signals happening. And we quantify these signals. So here with the arrows, you can see this nice PLA interaction and also staining of these biomarker cells, proteins. And when you quantify this, we saw that the contacts of PD-1, PD-L1 were particularly established by CD8 positive T cells and in CD11C positive dendritic cells, whereas only CD68 positive macrophages were barely associated with T cells. Here you can see a figure of the percentage of cell-cell interactions according to the color of the biomarkers as a result of this analysis. And we also found a way using free software which is available on internet for all researchers, which is called QPath, to do an automated proximity detection. And this one is still in development, this algorithm we're writing, but we can already show some first results. So here you have the PLA the PLA picture of single PLA staining. And when you use a cell detection method, you can see that it can quite nicely already recognize where all the cells are. And when you use the pixel classifier to detect DAB stained areas, you can see that it can nicely find where all the PLA detection takes place. When you add this together, you can have a cell detection with the pixel classifier. You can make them different colors so that the computer can recognize what is what. Eventually, you can also make your PLA positive cells as an object, segmentate them and analyze them so you can get some nice percentage of cells out of your image. So you can also compute the depth intensity features. Here we have the picture again. So we have PLA positive cells, which are black, and we have PLA negative cells in red. And we're using the QPath's built-in cell segmentation algorithm to calculate intensity features based on the intensity of the depth staining in the cytoplasm. So here you can see that. And here is one of the tools you can use to analyze this. Of course, we are now trying to detect immunomarkers as well with the proximity detection in it. It's working already quite well. Here in black, the PLA in reaction, and in red you can see PLA negative cells again, where in blue all the nuclei are stained, which are not positive for any marker. So in our study, to come back to the cell cancer paper, we saw that PD-L1 alone, however, was expressed in the tumor tissue, showing, but the interesting thing was that in the PLA we didn't see this reaction. So this can sort of suggest that the PD-1, PD-L1 interactions maybe do not take place in the primary tumor, but maybe all the action is happening in the most close nearby lymph nodes. So some concluding remarks from the study is that our findings implicate that tumor draining lymph nodes as a key orchestras an anti-tumor T-cell immune response that can be induced following a blockade of PD-L1 in the tumor draining lymph nodes. And our data, or this data, is challenging the current dogma that PD-1, PD-L1 checkpoint act primarily at the effector tumor site and offers an additional avenue for biomarker and combination immunotherapy discovery. So hopefully we can investigate much more. So in a short summary, I hope that I give people some, I give everyone, the audience, some knowledge on how to set up a discovery proximity detection assay using the Roche DISCOVERY ULTRA platform. And that we have some knowledge about how to add the chromogenic multiplexing to the proximity ligation assay. And that PD-1 interactions in tumor draining lymph nodes, but not in tumor, correlate with prognosis in melanoma in this case. And who knows in what kind of tumors we can find this as well. And I would like to thank Dr. Jan van der Tussen from the Department of Pathology, who is the pathologist in this study, and the Department of Pulmonary Medicine, and Floris Dammeyer and Mandy, who are the co-shared first authors of this nice paper, where we can use the patient materials to study this PLA interaction. And of course, I would like to thank Margie, Angela, and Melanie from Roche for their nice interaction on developing this technique on our DISCOVERY ULTRA platform.
Thank you, Dr. van den Bosch, for the wonderful presentation and for helping to shed some light on a topic that's of such interest in the research field of pathology and oncology. There have been several questions that have been submitted. Just as a reminder, please keep your questions coming in. If we don't answer them during this recording, then we will answer them afterwards and reply via email. Let's jump into some of the questions. So, Dr. van den Bosch, do you think proximity detection would be a suitable tool for future diagnostic use?
Thank you for the question, Mark. I think this technique will be a very suitable tool for the future in diagnostic use. One is because we show already in this melanoma study that it can be differentiating between survival and survival data of this patient. Maybe if we study this more in more tumors and we can find a more strong correlation, it can be used as a tool, just like PD-L1 is used now as a therapeutic tool. And the other point is that when you transfer the PLA detection kit to the ultra, to the benchmarks, so the diagnostic machines, I think that the PLA kit is not a very large kit, so it can fit into the benchmark machines very well. So I think it is very useful. Yes.
Okay, great. I've got another question for you. So how long does the proximity detection run actually take? And as a follow-up, how long does it take when you add chromogenic IHC on top of it?
So the PLA run itself only with DAB takes around 8 to 9 hours. Depending on your primary antibody of interest. So with 60 minutes incubation, 8 to 9 hours is how long it will take for one PLA reaction. If you add chromogenic immunohistochemistry, so you have to take into account the antibody denaturation steps, which every time takes 24 minutes, and the incubations, it can add up to eventually, in our case, 20 to 23 hours. Yeah.
Okay, thank you. Another question, what are the most important parameters you need to take into account when adding your IHC biomarkers?
So one of the most important thing is the antigen retrieval step you need to take in account, and also the marker, the localization of the marker you like to stain. Like you always need to do with chromogenic multiplexing is check whether you have nuclear markers, cytoplasmic or membrane markers, and you need to take into account which one are overlapping and which one are not. When they're overlapping, you need to choose your translucent chromogens on your run, and you also need to see if you have strong or not strong epitopes, so that you can choose to do them in the first or the last sequence of your run.
Okay, thank you. Another question for you. So, are there any considerations when you're actually mounting your slides? Do you have to do any pre-treatment before cover slipping?
So for now, what we do is we do the same as when we do a normal chromogenic multiplex or or DAB stain. We wash the slides in. First, we choose the slide cleaning in the discovery procedure, and after the slide cleaning, when the run finished, we also wash them in just normal soap in warm water. And then we cover slip them. In our case, we cover slip them in the HE600 of Ventana, in the cover slipping procedure. But you can also cover slip them with anti-fading medium, so something like Ismol mount or anti-fading medium to keep your chromogens nicely. Yeah.
Excellent. And then I think just one last question. You know, with the size of the assay, with all the different reagents that are being used, how do you solve the problem when the carousel is fully occupied with the reagent dispensers?
That is a good question. One of the things we do for that is to put the primary antibody step on manually, bring it up manually. So, for example, you can save 3 spots if you manually pipette the 3 primary antibodies after the PLA. But you can also make your detection open, so you can use yellow, for example, to add your yellow separately to the slide as a manual application step. Yeah, this is the way you can solve the problem with the carousel. You can take some of the steps out, which you can also make it manually. Yeah.
Okay, great. Thank you for answering those questions. We've come to the end of the webinar for today, and I'd like to thank Dr. van den Bosch for his excellent presentation and for the audience for logging in and joining us today. As a reminder, please continue to send in your questions, and we'll make sure we'll get your answers back to you via the emails. And also, just as a reminder, this presentation will remain available to view on Labroots on demand after today's recording. Thank you for joining, and we'll see you next time.